Bueno, buenos días. Si no os importa, empezamos ya porque son las 12. ¿Se oye bien? Se oye, ¿verdad? Es un placer para mí presentar a Pablo Palacios. Trabajamos en el mismo grupo a pesar de que está como investigador distinguido en el Instituto de Ciencias Agrarias. Pero tenemos mucho que ver desde el punto de vista temático. Trabaja en relaciones planta-suelo y en intentar entender la diversidad funcional y la diversidad de microorganismos en el suelo y su papel en la respuesta de distintos ecosistemas al cambio climático. Pablo hizo su tesis, su PhD en la Universidad Rey Juan Carlos con Fernando Maestre, recibió el premio extraordinario, disfrutó de una beca Fulbright en Estados Unidos, en Colorado State University, y una Marie Curie en el CNRS de Montpellier. Allí trabajó sobre todo sobre la descomposición de la hojarasca y su papel en el ciclo del carbono en distintos bosques, y fue cuando se reincorporó en España con Juan de la Cierva de la Universidad de Nuevo que empezó a trabajar más en sistemas o a interesarse más por sistemas agrícolas y el papel que tiene el suelo para la productividad de distintos cultivos. Después tuvo una Ramón y Cajal, con la cual se incorporó aquí al Instituto de Ciencias Agrarias y ha tenido también una beca Leonardo del BBVA, así que ha tenido muchas cosas y tiene un currículum increíble con unas contribuciones muy importantes en el campo que han sido publicadas en Nature, Science, Penas, etcétera, etcétera. Puedes mirar su website. Y hoy nos va a hablar sobre evidence for large microbial-mediated losses of soil carbon under anthropogenic warming. Muchas gracias, Pablo, cuando quieras.
Bueno. Me habíais dicho dar la charla en inglés, ¿verdad? Okay, well, thank you very much, Anna, for the nice presentation. And thank you also, of course, Rob, for the invitation to be here. It's really a pleasure, and I feel very honored to be giving a talk in this amazing room in the Natural Science Museum, which is a museum that I like it so much that I've been coming here since I'm a child. And now that I have two children, we come very often, so it's very special for me to give a talk right here. So thank you also for all of you who came here to listen today, and for those of you that are attending online. So today I'm going to talk about the interactions between the climate system, soil carbon, and soil microbial communities, because global anthropogenic warming is expected to increase soil carbon losses to the atmosphere, reinforcing climate warming. Okay, before starting the talk, I would like to thank all the amazing network of collaborators that have contributed to shape my views on this topic over the years. A bunch of ecosystem ecologists, microbial ecologists, and ecosystem modelers that have really helped hand by hand with me in all of the results that I'm going to show you today. Well, the carbon cycle feedbacks is a very important feature when it comes to predict climate change, because there are a lot of interactions between the atmosphere, the land carbon, and the ocean carbon that interact with climate change to contribute to the concentration of CO2 levels. Well, in the graph below, you have different representative concentration pathways. If you move from left to right, you have from less technological advance and less political actions, so more emissions. Of course, there is a lot of climate sensitivity towards these emissions, but when you incorporate the carbon cycle feedbacks into these predictions, these are for the 2100, of course, the range of variation increased quite a lot, especially in the upper level. When you try to transform these emissions into CO2 concentration in the atmosphere, this is a more tricky task. I'm not an expert in this task, so I'm going to just want to show you how it looks like. In black, you have the reference of the CO2 levels expected by IPCC. In the IPCC, I don't know where it's going. Okay, in the IPCC, they actually incorporate just the single best scenario for climate change. But what about when you incorporate a lot of potential outcomes? So all the black points are an ensemble of models, And you have two different Earth system models that incorporate the feedbacks. So the range of variation is increasing also in the upper level quite a lot. And it can change a lot the pictures when you only consider the anthropogenic sources of CO2 emissions. So today I want to focus on the land carbon climate feedback, and especially in soils, because it's my field of expertise, and because, of course, they are really fun to work in. And they represent the largest carbon pool in terrestrial ecosystems. So they store more than the atmosphere and the plant combines, and you can find these huge amounts all over the different biomes on Earth. Of course, you have cold ecosystems where plant growth is very small and there is a lot of carbon in the soil, but even in tropical ecosystems where net primary production is huge, there's a lot of carbon incorporated also in the soil. And the soil is also responsible for different ecosystem services, from food security, soil fertility, erosion control. And well, with this, I want to mean that there is a lot of co-benefits in preserving and incorporating soil organic carbon through time. Well, in the graph below, you have a very simple representation of the biophysical soil carbon cycle, which is just a product of the inputs and the outputs. So you have carbon gains, of course, from photosynthesis that are incorporated in the soil, and then you have carbon losses through the decomposition of the soil organic matter by soil microbial communities. So this net soil carbon is annually balanced, and climate warming may disrupt this balance, because soil respiration is actually more temperature sensitive than plant photosynthesis. So climate change can alter this picture, and this is what we call the land carbon climate feedback. So the higher the temperatures, the higher the CO2 emissions from the soil, more atmosphere CO2, and that would mean more global warming. However, this has been studied in modeling studies and empirical studies over the last 20, 30 years, but there's still not consensus on the magnitude of this feedback, whether these soil carbon losses will equate to large or small CO2 emissions to the atmosphere. And all this lack of agreement, this lack of consensus in the literature can undermine our efforts to push to the politicians to incorporate these carbon cycle feedbacks into the climate policy negotiations. So what is the source of these magnitude uncertainties? Where are they coming from in the literature? Well, I have highlighted a couple of them. There are other ones. The first one is what we call the earth greening. I mean the earth greening caused by warming temperatures, not by CO2 fertilization. So increasing warming temperatures, it will increase the availability of soil nitrogen in the soil, so it will alleviate the plant nitrogen limitation for growth. So if you have more inputs to the soil, that it's higher than the soil respiration, that will equate to net soil carbon accumulation over time. So there's a bunch of papers that have been published with this over the years, but it's important to note that there is evidence, recent evidence, saying that this earth greening and the sink capacity of the land carbon may be slowing down, and also much of the uncertainty in the land carbon cycle estimation, it comes from soil respiration more than with plant growth. Another source of uncertainty comes from the variation of warming effects on soil respiration. As you remember, it's this flux of CO2 to the atmosphere. So there are contradictory results on how these effects vary over long time scales. You have long-term warming experiments that are experimentally manipulating warming in different ecosystems, and these responses go from initial increases, but there is also some studies that have found that this initial increase in CO2 respiration is attenuates over time. So it seems like the ecosystem may be adapting to these increasing temperatures. Two potential mechanisms has been hypothesized to be driving this response. One of them is the substrate limitation for microbial decomposition, which is just the depletion of the labile carbon sources with initial warming. Microbes, of course, prefer to decompose these labile sources, and there is no more energy for the next decomposition to go ahead. And also another mechanism is the microbial thermal adaptation. Well, I'm going to focus on this one because we have published a couple of studies on this regard in the last years. So these two sister papers that were published in Nature Ecology and Evolution in the same number, one of them was brilliantly led by Marina Dacal, which was a former PhD student in our group by that time. She went to Mark Bradford's lab at Yale University with a data set that we have put together incubating soils from different parts of the world at different temperatures. Mark has also collected a similar data set, but independent totally, and they were looking at the same pattern with two independent data sets. What they found was kind of surprising, and it was highlighted in a news and views. This is where this graph is coming from in the journal by Charlotte Alster, which I think it summarized more or less the design. So Bradford paper, it's a network of sites they are all LTR sites in USA, spanning boreal, temperate, and tropical ecosystem, and a nice gradient in mean annual temperature. Then the Dacal paper, it's a global drylands network, and also encompassing a wide gradient in mean annual temperatures. There are different hypotheses on how the soil respiration may respond to increasing temperatures. So you have the amplification or enhancement response. That's a potential hypothesis. You have the neutral, where the microorganisms are not adapting at all to changing temperatures. Or you may also have the compensatory hypothesis. In the compensatory hypothesis, the microorganisms from warm-adapted ecosystems will be respiring less to the same temperature level. So what we found in both studies, so here you have a plot with the mean annual temperatures of the different sites where soil samples were collected. Remember, these were incubated in soil. It was rapid soil incubations, and we got the potential soil respiration responses under an homogeneous microbial biomass to just control for all the potential driving factors besides temperature. So what you have here is, of course, soil respiration is increasing with temperature, but in the models where you incorporate the adaptation to the lab incubation, the response is actually much lower. And this was found in both databases. So this suggests, or at least is consistent with our theories on the physiological or biochemical adaptation of soil microbial communities. Of course, it's not a definitive test because soil respiration is a broad ecosystem response that includes individual, population, or community level responses, integrates all of them, so we cannot partition and look into the mechanisms. That would necessitate other types of experiments with, for instance, microbial growth responses or these kind of things, but we think it's a robust indicators of the potential for this microbial thermal adaptation to be found in natural ecosystems. Well, these sources of mechanistic uncertainty that I have told you about, earth greening on one hand, microbial thermal adaptation on the other hand, can definitely, well, undermine our perceptions of whether we have confidence or not in the existence of this land-carbon climate feedback, and especially on whether they are of a magnitude important to be considered when defining the climate targets. So to try to help move forward in this sense and try to summarize whether we have the existing evidence on this topic, we organized a thematic sessions in a British Ecological Society meeting a few years ago, trying to put together the vision of different researchers that were coming from different sides of the story and with contradictory views. It was kind of difficult task, but also very, very insightful, and we published these perspectives. So the first line of evidence doesn't come from our own research, but it comes from global databases on soil respiration. So this database, this map is from the SRDV database that is pulled together by Ben Lamberti from the national laboratories, and it's a massive database with more than 10,000 points. Of course, there is some geographical bias, as you might find in any global database, but it's a really valid resource. So what emerged from this database is that temperature is a major control on soil respiration rates. So we have a Q10 of 3. That means that with 3 degrees of increase in temperatures, you will have a triplicate in soil respiration. But of course, this is not a direct indication of soil carbon. It's only a flux responsible or affecting soil carbon. So what about the net soil carbon response? Well, for this, we gathered data from the EarthGrid database. So you have here the soil carbon density in tons per hectare across different mean annual temperatures. And what you can find in this graph is that soil carbon accumulates in cold systems. For instance, Arctic, subarctic, or alpine ecosystems store a much higher concentration of soil carbon across the whole soil profile, from 0.5 centimeters to even 200 centimeters soil depth. And you can find this across the globe. What is going on here? Well, a potential explanation, and of course, this is evidence from space for time substitution, so it's not the most direct evidence for this. But in cold systems, plant growth is really small, really low, but soil respiration is even lower. And that fact over the years and over millennia can lead to the accumulation of soil organic carbon decomposition, soil organic carbon accumulation in the soil. Well, what's going on in these areas? What's going on in cold systems for them to be, the larger biomes with more carbon concentration? So one of the important features in some of these areas is that they have permafrost soil. So permafrost soil is the soil that remains frozen all the year around. And they have special characteristics because, of course, the composition is temperature limited and the carbon can stay there for millennium time scales. For instance, the mean annual resident times of carbon in these systems can be up to 10,000 years. So this region can be found in Northern Europe, in Russia, Greenland, USA, Canada, and there's also an important part in the Tibetan Plateau, which is actually very climate sensitive. And there's a lot of carbon in these areas. What's going on with these areas is that climate change is promoting the melt of this ice. So these soils are spending more and more months during the year where this active layer, where the soil is not freezing anymore, or is only freezing a bit during the summer, this depth layer is going down in the last year quite a lot. And this generates the whole landscape that can be seen from satellites, where you have these gaps in between these tundra ecosystems where the soil just collapses, and you have these water points. So having this in mind, we wanted to address what is the type of carbon that is stored in these cold systems. So we know the cold systems store a lot of carbon. We know they are sensitive, for instance, in the permafrost soils. But there is also a lot of new knowledge on the type of fractions in the carbon that more or less drive whether this carbon is more sensitive or not to warming. Traditionally, this has been the approach of the biochemical recalcitrance. The biochemical recalcitrance states that it's the chemical composition of the carbon that drives its vulnerability to disturbance or to warming. For instance, plant residues with a lot of lignin can difficult litter decomposition, and that will mean more accumulation of carbon in the soil. This biochemical view has been contrasted in the last year with a new one, more focusing on the physical interactions between the carbon and the soil mineral matrix. So this emerging, say, paradigm of soil carbon persistence over time says that you have, on the one hand, particulate organic matter, So this would be more or less the results of undecomposed or partially decomposed plant debris and animals debris. The lifetime is quite low, and they cycle very fast. On the opposite, you have the mineral-associated organic matter, which is this matter that is mostly composed by microbial byproducts that are stored in interaction with the soil minerals or inside the soil particles, which are meaning that they are much more difficult to be able to be decomposed by microbial communities. So the lifetime is much higher, and they are slow cycling. Okay, so this is the approach we follow, and that we wanted to assess how is the carbon accumulation in the soil systems in regards to this new approach. So to do that, and this is ongoing work, we just did a meta-analysis. We went to the literature, and it was a lab task where all the people, many of them are around here right now, were investigating the different articles and extracting data. And we collect a nice data set of different sites in Arctic, subarctic, and alpine ecosystems to answer these questions, to see whether organic carbon in cold systems is dominated by one fraction over the other. So that's what we found. So what you have here is the organic carbon concentration in the two different fractions, in green the particulate fraction, and in orange the mineral-associated fraction. And the particulate organic carbon fraction has higher concentration than the mineral-associated. And you can find this either in permafrost or no permafrost soil. Well, there are different explanations to this pattern, and we are still digging on the potential mechanisms, of course, with limitations because it's an observational data set. But it seems that mineral-associated organic carbon saturates at around 50 grams per gram of soil. So of course, the availability of soil minerals is not infinite in soils. So it gets to a point where there is no more minerals available to interact with, and it seems you saturate the response at that time. And you don't have this issue with the particulate organic carbon, which is, of course, fed by the plant debris. So now we know that cold systems are dominated by the particulate fraction. Does this has an effect on the warming sensitivity or temperature vulnerability compared to other systems, milder biomes, for instance? So to do that, we collected experimental studies that have tested the effects of experimental manipulations in warming, either with infrared heaters or open-top chambers, on 3 different fractions of carbon in soil. So SOC, which is soil organic carbon, which is the bulk soil, and then the 2 fractions, the mineral and the particular. And we compare the response in the cold systems versus other kind of biomes. Well, it seems to emerge a pattern where warming is decreasing. So here you have an effect size. So the more to the left, it means that warming is decreasing carbon compared to the control plots. So it seems that soil organic carbon is decreasing with warming, and that decrease could be driven particularly by the temperature-sensitive particulate organic carbon, which agrees what we found, what other people have found in the literature. So to wrap up all these results on cold systems, so this is the situations we may have in cold systems where they are warming between 2 or 4 times higher or faster the global rate due to phenomena such as the Arctic amplification. They do have the largest soil organic carbon stock, which is stored in the most vulnerable fraction. So I think those are all the ingredients for what some people have said the Arctic carbon time bomb. And this affects all of us, of course. It's not an effect that will stay in the Arctic. No? What happens in the Arctic doesn't stay in the Arctic. I'm not an Arctic ecologist, but these are the kind of results that we always listen, when scientists are arguing to incorporate climate change feedbacks and the importance this to obscure, for instance, some of our efforts to mitigate climate change with, I don't know, soil carbon sequestration or tree plantations. These are the kind of things that you have to balance. Okay, so this is the last part of the talk. So as I told you at the beginning, there is a lot of uncertainty of the magnitude of this land carbon climate feedback. I hope I have convinced you right now that there is potential for this land carbon climate feedback to be important. But of course, we have to build confidence because there is a lot of uncertainty in some of its estimations, for instance, on the soil respiration. So here I'm going to describe an approach we have followed to how to move on in our research when you have to deal with such uncertainties. So these are soil organic carbon models. There are basically two families of models. The first one in the left is the classical first-order dynamics, where CO2 released to the atmosphere is proportional to the soil carbon inputs. And this kind of models that you may have listened, this is the kind of models that are used, for instance, in Century or Roth Carbon. They work really, really nice at regional spatial scales, but they sometimes have a lot of problems when predicting field observations from these modeling predictions. There's also a new set of models that incorporate all the emerging findings on the importance of microbes for soil carbon cycling, the microbial explicit dynamics, where microbes are allowed to feedback to the soil carbon decomposition through their enzymes. So of course, there are support for both of them in the literature. And what we try in a couple of recent papers is to say, okay, so when you don't have to support for the exact modeling in the literature, you can also hypothesize different microbial by temperature relationship, because there is a lot of potential outcomes for different positive or negative. For instance, this is carbon use efficiency by mean annual temperatures. You could work with soil respiration or with any other parameters you want. So how to move on when you don't have this consensus in the literature? So an approach would be this model data integration, where you can test different competing hypotheses, of course with biological support, different modeling structures, and test that against observed data in large spatial scale data sets along environmental gradients. And that's what we did in a couple of recent papers. And that's what we did in the perspective paper that I was talking to you a while at the beginning. So here you have two different carbon use efficiency by temperature relationship, a positive and a negative one. And then in blue, you have the first order model, and in red, the microbial explicit model. So it seems that the positive carbon use efficiency relationship, it matches the observed data in global databases. These are data from our own experiments in the DACAL paper. But what about soil carbon? Because of course, soil respiration is just a flux and it's not indicative of net soil carbon accumulation. So in the carbon models, unfortunately, we didn't have the data at that time to trace that, but we already found support for soil respiration for this positive relationship. So when you have this positive relationship, the two models give you different outcomes. So in the blue model, in the first order model, there is no response of percentage of change in soil organic carbon with MAT. I'm sorry, I haven't told you the percentage of change is respect to the baseline model, which is the black line, so we can compare both models on regards to this baseline. So the first order model doesn't seem to find any response at all. But the microbial explicit model actually finds a negative relationship between respiration and MAT. So if you remember, this matches quite well the latitudinal pattern that we found at a global scale in this relationship. And it could be that with higher temperature, you have higher carbon use efficiency, so that means more microbial biomass available to decompose carbon. It could be a potential explanation for this observational pattern. So more than this to be the definitive result at all, I think this provides a good approach to advance our knowledge when we don't have the strong support for one particular hypothesis or for one model structure. And yeah, this is more or less the end. So just want to remark that we think that we have enough evidence for large microbial-mediated carbon under warming. It's an important question whether these soil carbon losses to the atmosphere are of a magnitude enough to offset the human efforts, for instance, to mitigate climate change via natural climate solutions. But of course, we think this means that we may need more aggressive reductions on emissions to meet the similar climate change target if we consider this kind of feedbacks. Just very rapidly, I want to show you a few follow-ups that we think could be nice to reduce the uncertainty around this magnitude. So one of them would be, of course, to study soil decomposer communities, microbial and fauna communities in cold regions where we don't have a lot of information, and incorporate this information in soil organic carbon models. We also have to move beyond the Q10 coefficient, which is, of course, a limitation of the reality, and try to incorporate different physiological models, more complex one. You can also think about incorporation microbial functional information in carbon models, and of course, including other environmental factors that I have excluded in our analysis on purpose to make it more simple, such as soil respiration, soil moisture, or even plant inputs. And also a particularly important one that I think it emerges, at least is something that I'm not really sure how to do it, is when you have to communicate this kind of results that has a lot of surrounding uncertainty, how to communicate this to the politicians so they can capture the message. So I think it's some space for collaboration with social scientists that may give us some insights on this. Yeah, before ending, I'm sorry about this, I just want to do a little bit of auto-promotion because there will be some people in the room interested or watching online. But we are offering right now 4 postdocs in a recently funded project on biodiversity and ecosystem services responses to climate change in urban ecosystems. I'm leading the soil carbon part, so we will be hiring a postdoc here at the Institute of Agricultural Sciences, working on similar questions that I have shown you today, but on urban systems. So yeah, that's the end. Thank you very much for your attention.
Thank you very much, Pablo.
You're welcome.
Are there any questions? Joaquin has a question.
Well, thanks, Pablo, for the talk, for the impressive wrap-up you made about many of the things you've been doing about this feedback. Well, as you rightly said, there are some biases in the global data we've got, and there are some evident gaps in that. So I'm a bit curious about some of the saturating relationship you included. I'm curious about the outliers, okay? Do you have any hint about where are those outliers? Because it could be the case that those outliers that could be driven that relationship could be just in some of the ecosystems we have sampled, and there are another kinds of relationships.
Yeah, well, first of all, the saturation response is also that some people have found also in other ecosystems, so we have evidence for this to happen. It could be not the case in our case. So we haven't explored this in depth right now because this is a database we finished collecting a few months ago. But yeah, it could happen. So when you are dealing with a meta-analysis, there is a lot of heterogeneity in the data set. We of course have tried to isolate that with including different random factors, and we have methodological difference that we have been minimizing in the study selection phase of the analysis. But yeah, good suggestion. Yeah, we could be having that in those places where there's huge organic carbon concentration that may be driving that.
And what I was thinking of, if you included some random factors, I'm sure you have included, for example, the type of biome or type of ecosystem, etc. Are there differences in the intercept at least, or something like that, even if it's a general?
We have addressed the effect of biome, because we have 3 different biomes that we are using here, so Arctic, Alpine, and subarctic, and the response trends are pretty clear in Arctic and Alpine, but not that much in subarctic. So, and that agrees with what we know about this, because other papers, for instance, using the Lucas database, is a European soil database with a massive spread across the European continent, and they have also found this saturation response, but this works especially in those sites, in the colder sites where you have larger soil organic carbon. So the subarctic could be some sites are even more close to temperate ecosystems, and maybe the response is not that clear there.
Thanks. It sounds really amazing, actually.
Thank you.
And exploring that, I love latitudinal gradients. So exploring that along the latitudinal gradient and cold, warm, seems like a nice avenue to. Yeah. Thanks.
Thank you.
Okay, Anna?
Thank you. Thank you very much. I enjoyed very much your talk, because it was very close to. And I had 3 questions that I would like to ask you. One of them was that one of the pieces of work that you presented that is very interesting is the one in drylands. And I'm surprised that I wonder if you consider also moisture, because one of the major predictions in these areas is an important change in precipitation patterns and the amount of rainfall, and whether you think that would obviously modify very much the response to temperature.
So yeah, it's a good question. Yeah, I'm sure it has an effect. In our case, so we try to isolate some of those influences because it's a soil incubation study. So of course, moisture was constant, but you have all this set of covariates as it happens with the mean annual temperature that could be affecting. But these were considered. We also considered different soil parameters, soil texture, CN ratio, and they didn't emerge as, or at least because this is quite a result from a few years, so I don't remember that correctly. So they were for sure taken into account, and despite all that, mean annual temperature was still emerged. I mean, it was not, of course, the largest predictor in importance, for sure, but we just wanted to show that the response is conspicuous at a global scale, so that was our goal. No?
And have you ever considered, I think so, because I talked to Marina at the time that I was in not only considering the average, the mean annual temperature, but also the amplitude, the variance of temperature in each one of these places?
So not for that work. We just consider mean annual temperature because we thought it was the one that encapsulates the potential adaptation of soil organisms to the environment, to the thermal environment. In the other one, where we are using data with soil grids, so the one where I'm showing that soil carbon accumulates at cold systems, we try with different climatic temperature variables from Chelsea database, like the temperature range, the minimum, the maximum, and they were showing the same pattern, that's for sure. In the global drylands, we did not.
And the last one, thank you, is when you study in which form this carbon stores in very cold regions, this particular organic carbon and the mineral-associated organic carbon, have you also looked at the soil type that determines the amount of minerals that you have present in the soil?
We are addressing that with the influence of soil pH and soil texture, and they are influencing the response. So soil pH is negatively, if I remember correctly, decreasing, and the soil sand is increasing the soil carbon in both fractions. But I mean, we collected the databases with the minimum information that were shown in the papers, and we try to fill a little bit of the gaps with using global databases. So it's not that we can do wonderful things with that. So.
Thank you.
I've got a question, but I want to apologize beforehand for being really ignorant about microbial ecology or microbial biology. So when you heat the soil and you record an increase in soil respiration driven by the microbial activity, is it essentially that the existing composition of the microbes, it's just they're all kind of increasing the rate at which they carry out respiration, or are you essentially observing big changes in the composition of your microbial community, which are driving those changes because certain, I guess they're not species or strains or genetic compositions of microbes are responding more rapidly than others or adapting more rapidly than others. And so is the heating of soils throughout the world over these large scale and fine scale kind of climatic gradients going to lead to massive changes in the composition of microbial communities, as well as kind of the rates of respiration that you've shown to us?
Yeah, I think it's a very good question also, because one follow-up of that question would be whether those changing compositions are leading the community to adapt to these new climatic conditions and to dampen the response, the overall soil respiration response, or not, no? And the answer is that we don't know. So soil respiration is a broad ecosystem response that includes, it's an aggregate response, I would say, of different physiological level responses, species turnover, population level dynamics, and we encapsulate all of this and call it soil respiration. So it has a lot of drawbacks. I think there are different ways of trying to dig in what you said. We have not done it so far, but I could imagine that one would be to work with microbial growth rates with specific strains. So you can do that in the lab with culture methods. And you can also use compositional data that you have addressed with sequencing methods or whatever, and try to see whether that explains part of the response. And the studies do find that. So there are a lot of warming studies using open-top chambers, and they relate compositional change with carbon change. And for instance, one response that happens quite often is that the carbon after warming remains a bit recalcitrant, and you have communities that are adapted to the greater recalcitrant carbon sources. So they go in parallel.
So these kind of composition change could be related to kind of the pattern that you showed of where you get a sort of an attenuation of the carbon release, and then it sort of all gets released again as different strains are coming in and becoming more abundant.
I mean, of course, it could be happening. I mean, we talk about this microbial thermal adaptation, which is, of course, a very broad term as well. So we don't have the definitive test to know that with this broad soil respiration response. But we think this agrees with what others have said is the biochemical adaptations and how the enzymes respond to warming. But we cannot neglect this kind of composition and response to be going through at the same time, for sure.
Any more questions? Okay, so if there are no more questions, thank you very much, Pablo. Thank you for coming and making us use our brains more than we normally would on a Friday morning, I think. Okay, thank you very much, everyone, for coming as well. And see you next Friday for Ana Benite, I think, who's going to be a new Ramón y Cajal working in the museum, who has collaborated a lot with Borja Milá, for example, here, I think. And so it'll be exciting to find out what she's going to be doing here over the next 5 years. Okay, thank you, everyone.